This PowerPoint is on the cerebellum and the thalamus, and I'm going to break it up into two parts. The first narration will be on the cerebellum and the next one on the thalamus. So let's begin with the cerebellum, and I've purposely put this slide in here just to show you and again review a little bit about some of the things we've already done. Here is the cerebellum. This is connected to the brain stem by means of these three peduncles, the superior, the middle, and the inferior cerebellar peduncle. And by means of these peduncles, it's also connected to the spinal cord. So this is the spinocerebellar tracts, and it is connected to the cortex because the cortex sends fibers to a part of the brain stem, and from there it goes to the cerebellum. So you can see what a vast area of connections the cerebellum has. Let's move on to the next slide. We'll be coming back to this slide if needed. So let's move on to the next slide and look at the gross features of the cerebellum. And whenever you're looking at a slide, always compare your notes with the description and try to understand what's given. So the first thing is that the cerebellum is situated in the posterior cranial fossa below the tentorium cerebelli. And if you look at this image here, this is the ventral aspect and this is the dorsal aspect. And if I was to draw the cortex somewhat like this up here in this fashion, and this is the occipital lobe, the tentorium cerebelli lies here in this part. So this is the posterior cranial fossa, and you can see that the tentorium up here is separating the occipital lobe of the cerebral hemisphere from the cerebellum. So that's an important thing for you to remember. It is the largest part of the hindbrain, which consists of the pons, the medulla, and the cerebellum. So it's the largest part of the hindbrain. And here in this image you can see that this, again, as I said, this was the ventral aspect and this is the dorsal aspect. If you look here, the cerebellum is dorsal to the pons and the medulla, and we already mentioned it in an earlier presentation. It forms the roof of the fourth ventricle. So this part here is the fourth ventricle. So it forms the roof of the fourth ventricle. So it's lying posterior to both the pons, the medulla, and as it forms the roof of the fourth ventricle, it lies posterior to the fourth ventricle too. Now, just like the cerebral cortex, the cerebellum also actually has two hemispheres. So you have two hemispheres, a right and a left hemisphere, and they are joined across the center by a part which is known as the vermis. This vermis is clearly seen on the inferior or the ventral aspect. This is the dorsal aspect of the cerebellum. So this is actually what you're looking at here, the dorsal aspect. And if you were to look at this aspect, the ventral aspect, which is this one, then you can see that this vermis is more clearly seen. So this whole part here is the vermis. It's better seen on the ventral aspect where there is this deep groove present. So this groove over here, this groove is known as the vallecula. So there's a kind of a deep furrow in which you can notice this vermis. Now if you look at the gross features of the cerebellum, notice these lines which go across. These are deep fissures, and the area between each fissure is known as a folium. And if you were to look at it in a cross section, each little part here is a folium. This part here is a folium. So each little bit here between two fissures is known as a folium. Now the oldest part of the cerebellum is something which is known as the floccular nodular lobe. So this part here is called the flocculus, and it goes on anterior to form the nodule. And then the next part is an area actually present laterally, which connects it to the spinal cord. And then the most recent one is an area present here, which is called the posterior lobe. So we have, if we look here, we have the floccular nodular, which is actually connected to the vestibular apparatus. That's the oldest part. The next, which is connected to the spinal cord, it really lies laterally, but it's represented mainly by this anterior lobe. So that's the intermediate developmentally. And then the posterior lobe, which connects it, which is to the cerebral cortex and is also known as neocerebellum. This is the most recent part. Now when you look at the white matter of the cerebellum, obviously the gray matter, like the cerebral cortex, is on the outside, so it's known as the cerebellar cortex, and then the central area is the white matter. In the white matter, again, you have little islands of gray matter which are known as nuclei, and the most important nucleus is this dentate nucleus, which is present in the core of the white matter. And as I mentioned earlier, it is connected to the brainstem by the 3 peduncles. So let's look at the connections as given in your notes, and there are 3 main regions that the cerebellum is connected to. The central area is connected to the vestibular apparatus, and that is the flocculonodular lobe. Then the parts on the side is connected to the spinal cord, so this area is known as the spinocerebellum. And the part which is present laterally, represented mainly by the posterior lobe, that is connected to the cerebral cortex, and it's connected to the cortex through this. The cortex first connects to the pontine nuclei, and from the pons it goes to the cerebellum. And if you remember, here were the pontine nuclei present here. So we had the corticopontine fibers, and then we had the pontocerebellar fibers. So that's how the cerebellum is connected to the cerebral cortex. And again, here you can see that important nucleus called the dentate nucleus. The other nuclei are called vestigial and interposed nucleus. Now before I do the functions, I want to go over the internal structure and the connections, and then I'll come back to the functions. So let's look at the internal structure of the cerebellum. The cortex is divided into different layers. There are 3 main layers present in the cortex. The outermost is called the molecular layer. The second or intermediate layer is known as the piriform layer, and then the third is called the granular layer. Now in all these layers you have number of cells, all given different names. The most important cells that we constantly talk about are these Purkinje cells, which are flask-shaped cells. So these are the Purkinje cells, which are flask-shaped cells. And these are present in the piriform layer, and they are the only outputs from the cerebellar cortex. That means from the cerebellar cortex, these cells are the ones which send out axons, and these are the only ones which send out axons from the cortex, and these axons go out of the cerebellum. So that's what is meant by they are the only outputs from the cortex. Then we have fibers, and there are 3 different types of fibers. Intrinsic fibers are fibers which connect cells within the cerebellar cortex itself. So from the molecular layer, if you have a cell which comes and relays on the Purkinje cell, that's an intrinsic fiber. Then from the granular layer, again, if you have cells which come and relay like this, that again is our intrinsic fibers. So those are intrinsic fibers. Then we have fibers which come from outside, which are afferent fibers. Some of these you know. For example, you know the spinocerebellar tracts. So you remember the spinocerebellar tracts. Then if you remember, we also had something which was called olivocerebellar. From the inferior olivary nucleus we had fibers going to the cerebellum. And then if you remember, we had these pontocerebellar. So you know, these are some names which should be familiar to you. So these are afferent fibers, that means they're going to the cerebellum. And these afferent fibers are of two types. One are known, so these afferent fibers are of two types. One are known as mossy fibers, and all cerebellar afferents, that means all fibers entering the cerebellum from different sources, except from the inferior olivary nucleus of the medulla, all of those are known as mossy fibers. The second type is known as climbing fibers, and these are from that inferior olivary nucleus in the medulla. The olive, remember, the olive was because of this inferior olivary nucleus. So these are known as climbing fibers, and the reason they're called this is because as they ascend, as they go through the layers of the cortex, as they're going up like this through the layers of the cortex, they look like a vine on a tree, like a vine on a tree. So it looks like it's climbing a tree, so that's why they are called climbing fibers. And these climbing fibers come and synapse directly on the Purkinje cells. The mossy fibers, they go up. If you look up here, these are mossy fibers. As they go up, they synapse on different cells in different layers, usually in the granular layer. So they synapse on the granule cells. These granule cells will then go and synapse on the Purkinje fibers. So that's how the system is, and we'll look at that a little later. The efferent fibers are fibers which go out from the cerebellum, and they all begin as axons of the Purkinje cells, because remember I told you that these were the only axons which went out of the cerebellum, so they were the only output. Most of these efferent fibers, and if you look at this picture here, so if you look, here is a Purkinje cell, this is an axon, these are the dendrites. So the axon from the Purkinje cells, they go to the nuclei which are present in the white matter. So you can see they are going to this central nuclei. So most of them, they actually go to the cerebellar nuclei, and then from the cerebellar nuclei the fibers will go out finally. Some Purkinje cells some axons may go out directly, so they may not necessarily, a few of them may go out directly, but most of them actually synapse with these nuclei, and then the output goes out. Now the basic circuit is, you have mossy fibers which come in, they go and synapse on these granule cells. So these are the granule cells. So you can see these mossy fibers, number one, they go and synapse on these granule cells. These granule cells will send off little after, their processes will come and synapse on these Purkinje fibers. The Purkinje fibers then will go out and synapse on these central nuclei, and then from the central nuclei they'll go to the various parts. So this is for the mossy fibers. For the climbing fibers will go and directly synapse on the Purkinje cells. From the Purkinje cells, the output will go to this central nuclei, and then they will go out. So that is the way the system or the circuit works. Now the thing is, so if I was to draw it over here, so let me say that this is a Purkinje cell, and this is its axon which comes out, and this goes to, let me call it the dentate nucleus. The dentate nucleus again looks very, very similar to the olivary nucleus. So that's the dentate nucleus, and then from here cells go out this way. So this is the Purkinje cell. Now what happens is, all the impulses which go to the Purkinje cells, that means from the climbing fibers, so these are climbing fibers which synapse here, so climbing fibers which synapse here, and then from these granule cells which go here. So the mossy fibers, they go first to the granule cells, and then the granule cells go to the Purkinje fibers. So all impulses which go to the Purkinje fibers, as you can see, they are excitatory. So that means they excite the Purkinje cells, or they stimulate it. But the Purkinje cell itself, when it goes out and goes to the dentate nucleus, this actually inhibits the dentate nucleus or any other nucleus in the amongst the central nuclei. So its impulses are inhibitory. Now there are other cells which are present up here. Here we only talked of, two cells. We've only talked of Purkinje and we've only talked about these granule cells. But there are a number of other cells in the molecular layer and other cells present in the granular layer, and they interact with each other and influence each other. What they do is that they come in, and let me actually use a different page for that. So let's take a new page so that you understand how the cerebellum works. So let's see how this works. So let's say we have these Purkinje cells, and this axon of the Purkinje cell goes and relays on the central nuclei. So these are the central nuclei, and from here fibers exit from the cerebellum. So the influence of the Purkinje cell on the central nuclei is inhibitory, and these are the dendrites of the Purkinje cell. The climbing fibers come and synapse on this, so these stimulate it, and then we had those mossy fibers which went up, synapsed on certain cells here, these came here and synapsed here. Their influence also is to stimulate the Purkinje cell. So if these stimulate the Purkinje cell and then it carries out its action, what it will do is it will inhibit these nuclei, and then the output from the cerebellum would be inhibited. So you don't want that to happen at all times. So what happens is that we have certain other cells which are present in the cortex, and these cells, they come and synapse on the Purkinje cells, or some of them will come and synapse on these cells. Their influence is inhibitory. Now the effect is the effect of all of this is one side there is stimulation, on one side there is inhibition. So what happens is that they influence the Purkinje cell. So they influence how much the Purkinje cell is going to be stimulated or inhibited. With the result, the Purkinje cell therefore no longer always inhibits the central nuclei. It only inhibits at some times when it is stimulated. So if the Purkinje cell is not stimulated, then it cannot carry out its effect. So which means that the dentate nucleus is no longer inhibited and impulses can pass out. So this is what is meant by the inhibitory effect of the Purkinje cells on these nuclei is fluctuating, because it depends on the combined action of all these cells and these fibers and their effects. So that is what is meant by the fluctuating effect, and this helps to modify the activity of the brainstem and the cerebral cortex, and through that the muscular activity. So let's go back to our PowerPoint, and now let's look at the next slide where we are looking at important cerebellar afferents. So again, the numbers that you see here are numbered based on what you have in your notes. So the first one is you remember these, the posterior spinocerebellar and the cuneo-cerebellar fibers, which are carrying information from the muscles and the skin of the trunk and the limbs. So these are carrying information mainly from the muscles, not the skin really, mainly from the muscles. So these come in, and if you remember the posterior spinocerebellar, it passed out through the inferior cerebellar peduncle. So you can see this is number 1, which is passing out through the inferior cerebellar peduncle, and this will go up as mossy fibers. So it goes to the cortex. The second is the anterior spinocerebellar, and the anterior spinocerebellar, if you remember, it went all the way up to the to the midbrain, and then from the midbrain it actually crossed over. So I'm showing you this anterior spinocerebellar. If it was really on this side, it actually crosses over and then goes, because remember it has to go to the same side cerebellum. So number 2 is the anterior spinocerebellar. Just so that you understand, the posterior spinocerebellar went all the way and it went this way. The anterior spinocerebellar, which is carrying fibers, if it's going this way, it actually crosses over in the spinal cord, if you remember, and then it traveled on the opposite side. But when it reaches the midbrain, it recrosses and goes to the same side, because the cerebellar control is ipsilateral. The cerebellum controls the same side of the body. So by going to this side, you can see finally the cerebellum is still controlling this side of the body. So that's what happens. So that's number 2. Number 3 are fibers which go from the colliculi. So they go from the midbrain colliculi, and if you remember, the inferior colliculus was to do with auditory information, and the superior colliculus is a pathway in vision, so it is visual information. So from the colliculi, this area was called the tectum. So these are known as tectocerebellar fibers. So they go carrying visual and auditory information to the cerebellum. So they go to the cortex. So all of these again are mossy fibers. Then we have fibers which go from the vestibular nuclei. This is for balance, and this is the most primitive part. So from the vestibular nuclei, number 4, these are fibers which are going from the vestibular nuclei, passing through the inferior cerebellar peduncle and then going to the cerebellum. And then finally, from the pontine nuclei in the pontocerebellar, so the pontine nuclei in the pons, and the inferior olivary nucleus. So you have actually you have fibers which come from the cortex. So these are corticopontine fibers. So from the cortex, so if I draw the cerebral cortex, from the corticopontine fibers, and then the pontocerebellar going through the middle cerebellar peduncle, and these were the fibers which they will go to the part which is the most recently developed, because if I told you that the cerebellum, you had a primitive part, and then you had an intermediate part, and then you had a part which was called neocerebellum. So these are also known as neocerebellar fibers. Then the cortex also actually communicates with the inferior olivary nucleus. So from the cortex, you also have fibers which come down to the inferior olivary nucleus, and then from the inferior olivary nucleus, you have fibers which go across and they go to the cerebellum. These were the ones, this number 5, these were the ones which were known as climbing fibers. And why do we mention them specially? Because it is thought that the inferior olivary nucleus probably has a teaching function in the acquisition of new motor skills. So, that's why we mention it specifically. And also, this is one way that the cerebellum gets information from the cortex. One way is through the pons, and the other way is through this medulla. So the corticopontine, cortico-olivary fibers, and then pontocerebellar and olivocerebellar fibers. Now, having looked at these, what you should also know is that through which peduncle they are passing. So that's why I've drawn these 3 peduncles. So if you remember, the posterior spinocerebellar and cuneo-cerebellar, they are going through the inferior cerebellar peduncle. The vestibulocerebellar also going through the inferior cerebellar peduncle, because the vestibular nucleus is seen mainly in the medulla. And then obviously the olivocerebellar. So climbing fibers will pass through the inferior cerebellar peduncle. Through the middle cerebellar peduncle, the fibers will be pontocerebellar, because they're going from the pontine nuclei. And then through the superior cerebellar peduncle, you'll have the anterior spinocerebellar fibers which pass through, and then you'll have the tectocerebellar, because they're going from the tectum. So sometimes if you remember where the fibers are, it kind of helps you, or if you remember where the structure is, it'll kind of tell you which peduncle they'll go through. For example, if I told you pontocerebellar, So remember, ponto means it's in the pons. How is the pons connected to the cerebellum? Through the middle cerebellar peduncle. So therefore, pontocerebellar will go through the middle cerebellar peduncle. If I told you tectocerebellar, then you remember the tectum and this part, the tectum is also synonymous with colliculi here. I know there was a, it might be a little confusing sometimes, but remember the tectum and the colliculi, they are kind of, we use them synonymously sometimes. So from the tectum, which is present in the midbrain is connected to the cerebellum by means of the superior cerebellar peduncle, so that's how they'll go through. The anterior spinocerebellar, if you remember, that was one thing. It was spinocerebellar, but it did this crazy thing of crossing over and then going back like this. So you remember that was the anterior spinocerebellar. So it's something odd about it. So it goes through the midbrain. You see it all up to the midbrain. And how is the midbrain connected? Through the superior cerebellar peduncle. So that's how you're going to remember. Then for the inferior cerebellar peduncle, the posterior spinocerebellar, remember we never saw it up in the rest of the brainstem, we only saw it till the level of the medulla, so that means it goes through the inferior cerebellar peduncle. The vestibulocerebellar, the vestibular nucleus is seen in the pontomedullary junction, so the nucleus is seen mainly in the medulla, so it's going through the inferior cerebellar peduncle. And olivocerebellar, the inferior olivary nucleus seen in the medulla is connected to the cerebellum through the inferior cerebellar peduncle, and that's how it goes through. So that's how you're going to remember these cerebellar afferents, through which peduncles they are going through and where and what are they connecting. Now let's look at cerebellar efferents. So we're going to use the same picture, but now the direction is opposite. So here we have the Purkinje cells, which will send out fibers to these nuclei, and actually from these nuclei the impulses will pass through. So again, let's look at them. So the first one is that from the cerebellum you have a bilateral connection with the vestibular nucleus. So vestibular nucleus, if you remember, through the inferior cerebellar peduncle it sent vestibulocerebellar fibers. From the cerebellum, you have information going to the vestibular nucleus also. What does the vestibular nucleus do? This is important for balance. And once the information comes from the cerebellum to the vestibular nucleus, then the vestibular nucleus can send fibers down to the spinal cord as vestibulospinal cord, and that's how cerebellum controls balance. So you can see that's a bilateral connection. So this is now showing you the efferent fibers from the cerebellum to the vestibular nucleus, and obviously it will go through the inferior cerebellar peduncle, because if the afferents went through there, the efferents also will go through there because it lies in the medulla. The second is that from the cerebellum you have information going to the reticular formation. So this goes to the reticular formation in the pons and the medulla, mainly in the medulla. You have very few efferents traveling in the middle cerebellar peduncle, so that's why I just showed it to you in the inferior cerebellar peduncle. So for all practical purposes we'll say that the middle cerebellar peduncle had no efferents. So you have information from the cerebellum going to this reticular formation. From the reticular formation, impulses will go down as reticular spinal tracts, and these regulate posture and locomotion. So that's how again the cerebellum controls the spinal cord. So you can see from these two examples that I've given, the vestibular and the reticular, that the cerebellum is not going directly to the spinal cord. It's sort of making a pit stop first at the vestibular nucleus, and then here there's another one at the reticular nucleus. So it controls the spinal cord indirectly, or the lower motor neurons of the spinal cord, it controls it indirectly through these connections. And I'll go over this again. So that's the second one. The third one is here, where the cerebellum is connected to the red nucleus in the midbrain, and this is involved in motor learning. So we have fibers going from the cerebellum to the red nucleus, and they really go from the dentate nucleus. So these are known as dentatorubral fibers, and from the red nucleus they actually will travel up to the thalamus, and then from the thalamus they go to the cerebral cortex. So these are called rubrothalamic thalamocortical fibers. So this is showing you that to the red nucleus. And some fibers go directly to the thalamus, so these are known as dentatothalamic, and then from the thalamus they'll go to the cortex. So you can see they are going from the thalamus to the cortex. So you can see that information now, it got information from the cortex, but it's sending information to the cortex also. Now, one important thing to know about the cerebellar output is that what the cerebellar output, how it's conducted is, it is conducted to the sites of origin of the descending pathways. So if you see, this was the cerebrum, the brainstem, and then the spinal cord. So if you remember the origin of the descending pathways, so the descending pathways were from the motor cortex, so this is fibers coming down, and then we had brainstem nuclei. So you had fibers coming down from brainstem. So again, like for example, you had these vestibular nuclei, you had fibers coming down from here, you had the reticular formation in this area, so fibers coming down this way. So this is all this way. So what the cerebellum does is that its fibers, so let's say if we drew the cerebellum here, its output is conducted to the sites of origin of the descending pathway. So it goes, as you can see, it went to the reticular formation, it went to the vestibular nucleus, and then up here there was the red nucleus. So it went to the red nucleus, and then so it went to the red nucleus, and it is also connected to this, but it's connected to the cerebral cortex in an indirect fashion through the dentate and then going up, or to the red nucleus, and from the red nucleus up here. So you can see the cerebellar output is conducted to the sites of the origin of the descending pathways, and then these descending pathways you can see come down, and they influence the anterior horn cells or the lower motor neurons in the spinal cord. So what happens is that in this respect, the cerebellum, if you notice, there are no red fibers coming directly. The cerebellum has no direct neuronal connection. So there's no direct neuronal connection with the lower motor neurons, but it influences the lower motor neurons indirectly through the cerebral cortex and through the various nuclei in the brainstem. So that is important for you to know. And it does this, as you can see from here, it will do this. This influence is ipsilateral, because as it influences these motor cortex fibers, remember these will cross over and come down. So the left cortex is influencing the right side, so you can see that this indirectly influences the right side, and these fibers also cross over and come. So that's how the cerebellar control is ipsilateral. Let's now go back to our PowerPoint, and here we are going to look at the functions of the cerebellum. And what the cerebellum has been described as is, it's been described as a comparator. That means it compares the information it gets from the spinal cord and from the cerebral cortex, then decides, it processes that information and then sends out its directions to the cortex, and then the action can take place. So it compares what information it's, and depending on what information it's getting, it will act on that. So let's first look at this image here, and just to show you what I'm trying to show you is, so here's the cerebral cortex. So here is it's showing you a muscle and the muscle spindle. So let's say this is the spinal cord. So you can see that from the spinal cord you have impulses which go to the cerebral cortex. These are your sensory pathways. So this is the proprioceptive impulse. So obviously it makes a stop in the thalamus. and then from the thalamus it goes there. So this is information from the spinal cord going to the cerebral cortex, sensory information, and then the cerebral cortex controls the spinal cord through its corticospinal fibers. So you can see how the cortex is connected to the spinal cord through this pathway. Then the spinal cord is connected to the cerebellum by its spinocerebellar fibers. So you can see that these are sensory pathways to the cerebellum. So you have spinocerebellar pathways, but you have nothing going from the cerebellum to the spinal cord directly. So you can see there are no yellow. So yellow is, if you say, the motor or the efferent fiber. So there are no efferents directly from the cerebellum to the spinal cord. Then we have a reciprocal communication between the cerebellum and the brainstem. So, I gave you examples like the vestibular nucleus going across like that. So this is, and you have, and from the cerebellum you have information going to the brainstem. The brainstem connects with the spinal cord, and from the spinal cord we have fibers going to the brainstem. For example, you have olivospinal fibers, vestibulospinal fibers, reticulospinal fibers, and then you'll have information from the spinal cord also going to the brainstem. For example, you have spino-olivary fibers, spino-reticular fibers. So you can see there's again a reciprocal communication between the brainstem and the spinal cord. And then we have these fibers here where the cerebral cortex communicates with the brainstem, and these are, for example, the corticopontine fibers and the cortico-olivary fibers. And then from the brainstem, that same information now is passed to the cerebellum by means of pontocerebellar fibers or olivocerebellar fibers. So that's how the cerebellum gets information from the cerebral cortex. It's receiving information from the spinal cord, and then it compares that information, and then after processing that information, it sends information to the cortex, telling the cortex what needs to be done. That it does through the cerebello. For example, you had fibers which went from the cerebellum to the red nucleus and then to the cortex, or from the cerebellum to the thalamus and cortex. Remember the dentatothalamic fibers, and then dentatorubral, rubrothalamic. So that's how it gives its information to the cortex. So this is the circuit, and you can see how the cerebellum, because it's controlling the cortex, then the cortex sends fibers down to the spinal cord. So that's how motor activity is coordinated. So you can see that the cerebellar influence is this, that it is coordinating the motor activity. Now let's see what all the cerebellum does. So the first is it's the maintenance of muscle tone, and when we say muscle tone, we mainly talk about skeletal muscle. And what it does is it controls this, it harmonizes the muscle tone, and it's done this way, and it permits voluntary movement such as walking, etc., to take place smoothly. And there is precise movement for precise duration. So that means the cerebellum prevents unnecessary movements, and it makes sure that the movement doesn't go beyond a certain limit. So that's why we say precise movement for precise duration. So the movement does not go on beyond a certain limit. So, it limits voluntary movement. So that is one of its function. It maintains posture and gait, and it does this through its vestibular connections and through its cortical connections. It coordinates and makes sure precise voluntary movement occurs, and this it does by comparing the output of the motor cortex with information that it's getting from here, which is what I explained earlier, and then it indirectly influences the anterior horn cells. So you can see what it does is it sends back information to the cerebral cortex to inhibit agonistic muscles and stimulate antagonistic muscles. Thus it limits the extent of voluntary movement. So that is very important. It limits the extent of voluntary movement. For example, if you reach across to pick up a book and you flex your elbow, you don't keep on flexing your elbow. Once your action is done, then your elbow starts extending. So this is done because the cerebellum now can inhibit the flexors and stimulate the antagonists, which are the extensors. So it limits the extent of voluntary movement. Otherwise, you'll keep on doing what you were doing. And we'll see what happens in cerebellar lesions. So this is one of the important things to remember. It coordinates reflex and voluntary activity, which is pretty much the same thing. And the planning of sequential movements and correction of movement errors, this is very, very important, because what it does is the lateral zone of the cerebellar cortex, that what is called the neocerebellum, this is concerned with the planning of sequential movements of the entire body, and this is able to assess if there is a problem in movement. For example, if a movement is intended and another one is done instead, the cerebellum will control it, or the cerebellum will correct it. So this is important. And in balance, in balance, now the cerebellum receives information from the vestibular nuclei and it receives information from the tectum. So here we mentioned this earlier. So it receives from the vestibular nuclei, this is to do with balance, and from the tectocerebellar, this is for visual and auditory input. So this is also important in cerebellar function. So the cerebellum is damaged, your visual and auditory input, some of it will not matter at all because it's not receiving that information. Let's go on to the next slide. And this is basically showing you the cerebellar and cerebral connections. So here you can see the green are afferents to the cortex and the blue are efferents from the cortex to the cerebellum. So let's look at this. So here we have these fibers which come from the motor cortex. So they are coming down, and these fibers they go down. These are the corticospinal fibers. Then we have these fibers which come from the cortex. They go to the, up here they are going to the inferior olivary nucleus, and then we have the olivocerebellar fibers. What I've not shown here is they also go to the pons. So these are corticopontine, and from the pons they go to the cerebellum. So those are the pontocerebellar fibers. So this is how it is. It gets its information from the cortex. It sends its information to the cortex through the dentate nucleus. So either it goes dentatorubral, rubral thalamic, thalamocortical, or it is dentatothalamic and thalamocortical. So that's how it sends its information to the cortex, and that's how it makes sure that if there is any errors, those errors are corrected. Let's now look at the signs of cerebellar dysfunction, and based on what we have just said, let's see what are the signs that we will see. If you remember, one of the important function was maintenance of muscle tone and controlling posture and gait. So if the cerebellum is damaged, obviously muscle tone cannot be maintained, so you will have what is called hypotonia, or the muscle feels floppy. So there's decreased muscle tone. And you can see up here a child is being lifted up. When you lift a child up like this, in a little infant, this is really a dangerous thing to do because you can actually dislocate the radius. But otherwise, anyway, when you lift a child up like that, you can feel a certain resistance. But in this case, you can see that this child feels absolutely floppy. So this is what is known as hypotonia. So there is diminished resistance to passive movement, and this is because the stretch reflexes, the cerebellar influence on that is lost. Gait is changed. So the person walks with a wide base, as you can see here. They walk on a wide base, and so that they can keep their balance. And the person is usually stiff-legged to compensate for loss of muscle tone. So they walk with very, very stiff legs. They tend to lurch towards the affected side. So if the right cerebellum is damaged, then they will be lurching towards the right side as they walk. Then we have ataxia. Ataxia, the meaning of ataxia means actually there is no coordination or without order. Without order. And the muscles contract extremely weakly. They're weak in their contraction. The person has tremors when they, you ask them to do something. When fine movements are attempted, you see that there is shaking, which is known as tremors, such as when they button their clothes or they're shaving or they're writing. And if you tell the person to, touch their finger to their nose or do the heel shin test, because muscle groups cannot work together, because remember there is the cerebellum controls antagonistic and agonist interaction. Because this is gone, or what is known as cerebellar guidance is gone, so cerebellar guidance is lost, then the movements are incoordinated. So, there is kind of decomposition of movement. So you'll find tremors in these people. And normally the movement is self-limiting. But in this case, these tremors carry on. Let's see the other problems that we have when we have cerebellar lesions. So we have this decomposition of movements, which is what I just mentioned, the finger nose test. As the person approaches the nose, you can find their hands start, the tremors appear, and these are known as intention tremors, because it looks like they are doing it intentionally, which they are not. And here in a child, you can see if you hold a finger or a toy in front of the child and ask them to touch it, you'll see that the child with incoordination is unable to do it. So they'll go all over on either side, and they'll never be able to go close to what you want, and that is known as past pointing. Then we have dysdiadochokinesis. This is, you cannot do alternating movements rapidly. For example, pronation, supination. So they can't do this. The tendon reflexes are very slow. You have what is known as pendular knee jerk. That means, you tap the knee, and usually you should have extension, but this goes on for some time. And the reason is, normally the movement is self-limiting because of the stretch reflex of antagonist and agonistic muscle. But here, since cerebellar influence is lost, the reflexes continue as a series of flexion extension of the knee. Because, if you remember, I told you cerebellum normally limits voluntary movement. So if the cerebellum is damaged, obviously this is gone, so the movement keeps continuing. So that is what is meant by pendular motion. And then we have nystagmus. Normally the posture of eye muscles depends on normal functioning of visual pathway. So the visual pathway, where the eyes view the object of interest, and normal functioning of the eye muscles is also dependent on a more complicated path that involves the labyrinth or the inner ear. So the inner ear and the vestibular nuclei and the cerebellum. So if the cerebellum is damaged, obviously the eye muscles are incoordinated. So you have what is called nystagmus. And just to show you how nystagmus looks, I'm going to just change this. And notice in this picture, you can see how these eye muscles are moving, side to side. So notice, so you'll find this person, the muscles will go across from side to side. So notice how this happens. It goes across from side to side. So this is nystagmus. Then speech, because the cerebellum is important for muscle coordination, there is dysarthria, means there is difficult articulation. So the articulation is jerky and the syllables are often separated from one another. The speech is slurred and explosive. For example, they'll say something like, if their name was Anita, they would say, Anita. So they shout like that. So, and this is because of faulty coordination of impulses which reach the lips, the tongue, the palate, and all of that which controls speech. So, that's what is seen. So explosive and scanning speech, that means the speech is kind of broken up. And in the main thing that you see in cerebellar lesions is that there is no paralysis and there's no loss of sensation. So there are no sensory changes. So there's no paralysis and no sensory changes. And even though the muscle contractions are weak and the patient fatigues easily, there is no atrophy of muscles. That again is another important thing. There is no atrophy. So there is no atrophy because the corticospinal tracts, remember, they are fine. So these corticospinal tracts, they come down and they act on the anterior horn cell. So these guys are fine. They are just not getting the input from the cerebellum. So that's why there's incoordination occurring. But it's only atrophy will only occur if this final pathway is gone. So this is okay. So there is incoordination. The muscle contractions are weak because this influence is not there. The patient fatigues easily because of this, but there is no atrophy, no sensory changes, no paralysis. So this is important to remember in cerebellar lesions. I'd like to show you a person who has cerebellar dysfunction and the intention tremors. So let's move on here.
Tremor disorder, in addition to rest tremor and postural tremor, is intention action tremor seen with cerebellar disease. In this patient, who has had a traumatic hemorrhage involving one side of his cerebellar white matter pathways, he has a normal function on the right side, but the left side shows significant dyssynergia and intention tremor. This type of tremor contrasts, however, with the postural tremor, in that both the trajectory and the endpoint are abnormal, and most of this patient's dysfunction is in fact right at the endpoint when he tries to touch his nose or to bring his finger totally out. The trajectory is not normal, but the trajectory is not the area of maximal dysfunction. This type of tremor can also be seen in multiple sclerosis and other primary cerebellar disorders.
So you can see in that person when they were trying to touch their nose with their left hand, how the tremor started, and it was difficult for them to touch their nose with their on that side because there was probably a cerebellar lesion on that side. So I'll stop here with this presentation and then we'll do the thalamus next time.